Hello, everyone. Thank you for joining us for our webinar today, hosted by CIRM and Synthego. I'm Jennifer Bennett. I'm the Product Marketing Manager at Synthego, and I'll be moderating the webinar today. Recently, Synthego was named as an industry resource partner for CIRM, the California Institute of Regenerative Medicine. The Industry Resource Partnership positions Synthego amongst a small group of prominent suppliers to provide services, technology, and expertise in support of various discovery, translational, and clinical stage research projects focused on iPSC-derived cell therapies. Here to speak a few words about CIRM is Dr. Lisa K. Lisa?
So what I want to just say to you today is really briefly, I want to thank you, Synthego, for putting on this informational webinar. And I wanted to just highlight a couple of points from the CIRM perspective, which is first, acknowledging that CIRM, of course, does fund gene therapy approaches, including products made with CRISPR-Cas9 gene editing. And second, we have a funding pipeline, which is the slide I was going to show you. It ranges from discovery through translational and IND-enabling pivotal studies and into clinical trials. So as our grantees move from research all through the clinic, the question is often asked, which grade of reagent and quality of reagent is needed at each stage. So I'm really hoping that today it'll be helpful for both the CIRM staff and for our grantees to hear about your different offerings and which ones you recommend for the various different stages of development. And with that, I will turn it back to you because I know we're all interested in hearing your presentation.
Thank you, Lisa. So before I introduce today's speaker, one housekeeping function, we will have a Q&A session after the webinar. So if you have questions during the course of the webinar, if you want to please address those questions by putting them into the Q&A function, not into the chat, but the Q&A function, and we will answer as many as time allows. And if we don't, we will get back to you with an answer to your question. So speaking today on enabling GMP production of sgRNA for CRISPR-based cell and gene therapies is Dr. Kevin Holden. He is the head of science at Synthego, a senior member of the team responsible for developing platforms that deliver high-quality, scalable CRISPR genome engineering solutions. He helps to define Synthego's scientific strategy and innovation through collaborative engagement with key opinion leaders in the genome engineering community. He has over 15 years of biotechnology experience with a focus in synthetic biology and genome engineering. Thank you for speaking with us today, Dr. Kevin Holden, or Kevin, and take it away.
Okay, all right. So thank you, Lisa, and thank you, Jen. It's great to be here, and so excited to talk to you all about how we've tried to start enabling CRISPR-based gene and cell therapies through the development of a series of reagents that accumulate with a GMP type product. Let's say, okay, so as both Jen and Lisa mentioned, we have now entered a sort of closer relationship with CIRM, Synthego has, and so just to re-up that, we are now a industry resource partner for CIRM with the shared goal of helping to accelerate gene and cell therapies, specifically in our case with the use of CRISPR. Okay, so just to kind of take you through the outline of my presentation today, I'm going to be telling you a little bit about how we can help to enable scientists and awardees of CIRM grants to take them from the lab to the clinic using the CRISPR technology. And I want to tell you a little bit of, about three kind of aspects of that. The first is engineering, and actually how we've been able to build a CRISPR genome engineering company that supports cell and gene therapy developers from the bench to the clinic. I'll talk about how we actually deploy those technologies, specifically how we've developed what we call phase appropriate CRISPR guide RNAs that provide an end-to-end solution for cell and gene therapy. And then lastly, and the focus of most of the talk will be about how we help developers of cell and gene therapies in the CRISPR space interact with the regulatory bodies. And included in that will be some trends that we've seen and feedback from phase 1 clinical programs we supported. And then how we're helping to actually guide some of the regulatory bodies like the FDA towards understanding and characterizing fully these guide RNA molecules that are an essential part to developing a CRISPR-based cell and gene therapy. Okay, so just a little bit of background. We are a California company. We're based in the San Francisco Bay Area. So it's fantastic to be able to partner with the California Institute of Regenerative Medicine. Just taking you back a little bit to 2012 is when the company was first formed, and our goal was actually to try to enter the CRISPR gene editing space. And so the company was founded on kind of engineering principles of building a new type of RNA synthesis architecture. And by 2015, we had decided to focus particularly on trying to make what's called the single guide RNA format of a CRISPR guide. Up until this point, most people were using these molecules in plasmids, or they were trying to make them in the lab using in vitro transcription. And this actually leads to problems when you're trying to engineer cell lines, particularly with consistency and sort of like, I would say, cellular reactions to RNA molecules that are not purified. And so by 2016, we actually were the first company to launch what we call a discovery scale molecule. So this is a small scale molecule that's used for research use only. We decided to really make this an easy thing for people to order on a website, and also to how to analyze their CRISPR editing. We developed a tool called ICE. There was a publication on this. And this really helped to drive sort of the mass adoption and democratization of the CRISPR technology in the laboratory. And this is because we were providing a reagent that people could just order, just like you could a DNA primer. And it would show up in your lab in a couple of days. It's pretty cheap. And then you could use it directly in your experiment, and then you can analyze your CRISPR results. And we also started to learn how to use how to synthesize different types of RNA molecules as well. So those could be molecules that are not just protected on the ends of the molecule, and those protections help to stave off any sort of like immune responses that can happen in primary cells, but also kind of help to keep the RNA around in the nucleus a little bit longer. But we also started to learn how to synthesize guide RNAs that are heavily modified, and people are typically using these now for more in vivo applications. Learning how to make longer guide RNAs that people are using for different types of gene editing modalities or novel nucleases, and then even making hybrids with RNA and DNA. And so we kind of shifted away from just making reagents into also learning how to provide CRISPR as a service. So we decided to embark on a long journey to develop a cell engineering service. And we came to the point of what we call industrializing genome engineering. So we built these workstations and work cells that try to automate many of the processes that people use for gene editing. And so this was a sort of like a factory that we launched that enabled people to build cell models, for example, for disease in immortalized cell lines or iPS lines, so they could actually build a model of their disease and maybe perhaps differentiate that into a cell type or an organoid of choice. So this is something that we feel kind of goes hand in hand with supporting scientists as they move on this journey from understanding, what genes are interested in targeting to actually understanding what a model of their disease can look like. And then so we decided early on, after we started supplying discovery scale molecules, that we wanted to support scientists from the bench into the clinic. And so this led with the culmination of our first clinical GMP guide RNA factory that was launched in 2020, actually during the pandemic. This was the world's first GMP facility that was specifically designed just to synthesize CRISPR guide RNA. And so just to give you an idea, when I talk about industrializing genome engineering, we really do have a lot of experience now working with CRISPR-based molecules and with using the CRISPR technology itself to engineer cell lines. So since we launched commercially in 2016, we've synthesized more than 2 million molecules, individual molecules. Every quarter, we're typically synthesizing around 6,000 guide RNAs. We take on about 1,000 cell engineering projects for customers in industry, academia, medical centers per quarter. And since we've launched, we currently have about 1,500 citations of the use of either our products, analysis software, cells in the primary literature. And from the GMP perspective, since we launched that factory in 2020, we've manufactured over 100 GMP and GMP-like CRISPR guide RNA batches for clients. And so because of this huge demand that we were starting to see in the GMP space for clinical grade material, we decided almost immediately to embark on building a second factory, which we call Factory 2. And so our GMP Factory 2 is also located in the San Francisco Bay Area in Redwood City. This opened just this past May. And just on the right here, you can see Fyodor Urnov from UC Berkeley IGI, who came and was our speaker for our opening ceremony. This was a $28 million investment that we made in this facility. It's 18,000 square feet, and it's fully compliant for GMP. It's fully compliant and certified for GMP manufacture. It's a building that we designed and engineered ourselves, and it allows us to 20x the current capacity we have for making GMP molecules. And this really can significantly reduce the wait time that people have for obtaining GMP materials for their IND enabling or phase 1 studies. And so both of the facilities, the original facility and this new facility, have gone through 20 successful audits to date by clients. Okay, so why did we make this bet and spend all this money to build these expensive and complicated GMP facilities? Well, cell and gene therapy is the development of it is moving towards CRISPR now. And so about 70% of new gene editing trials are utilizing CRISPR. So people are trying to move away from using, for example, lentivirus and seeing how they can actually engineer cells either ex vivo or in vivo. And so there are currently 4 CRISPR trials that are in phase 2 and 3, and the data coming out from those is from CRISPR Vertex and Intellia is very positive. There's been over 50 new INDs that utilize CRISPR just this year alone. It's a market that is expected to grow to about $10 billion by 2030. And to date, there's been at least 200 people that have been dosed with CRISPR-enabled therapies. Recently, Exicel, which is the CRISPR Therapeutics Vertex treatment for both sickle cell anemia and beta thalassemia, and ex vivo CRISPR gene therapy, was recently given approval in the UK by MHRA, and it very shortly expected to be given approval by the FDA as well. So we fully expect once this therapy is approved by the regulatory bodies that we'll start to see an even greater explosion of the use of CRISPR technology for cell and gene therapies in the clinic. Okay, so I'd like to shift gears a little bit and talk about how we think about generating guide RNA molecules that are appropriate to each phase of the cell and gene therapy development pipeline. And so what we've done kind of based on feedback over the years from our customers and clients is to develop what we call phase appropriate guide RNA. And so we're looking to be a partner for people developing therapies all the way from when they first start their discovery into their preclinical and IND enabling studies, and then into the clinic. And so, like I mentioned, we have developed a suite of what we call phase appropriate guide RNAs. So this means first in the discovery phase, people will use research use only material. So this is material that is made at a small scale, is not ultra purified, but is pure enough to do very successful gene editing in any cell type, including primary cells. Then we have what we call INDE or IND enabling guide RNAs, and these are of greater quality and also manufactured under greater specifications and with kind of limited documentation. And then we have the full GMP guide RNA, which of course is synthesized in a closed environment with all the appropriate documentation required for the regulatory bodies. And so in addition to supplying phase-appropriate guide RNAs, we also work with our clients on their IND submission. So this can include helping with pre-IND filing and submissions, and also regulatory support and comments from the regulatory bodies after submitting an IND. So this is something we've decided to take on, especially to help our clients on this journey. And part of the reason is, the CRISPR technology has been really democratizing in terms of people being able to start to do things like knock genes out or tag genes or make single nucleotide variants in the laboratory on their own. And previously the technologies that were used to do these types of experiments and engineer cells in these ways were quite cumbersome and difficult to use and not very efficient. And in the same way that CRISPR has really kind of democratized how people are doing research in the lab, it's also allowed a greater number of people to actually sort of dive into this idea of developing a therapy for a disease, a gene therapy for a disease. Much more researchers than were possible before the CRISPR technology was around. Okay, so as I mentioned, just thinking about this sort of ladder, this continuum of guide RNAs, and as you kind of move up this ladder from discovery to toward the clinic, we're increasing the amount of quality that is associated with that molecule, and also the documentation that comes with it. So the research use only is sort of the screening scale. This is what you can buy on the website, just sort of directly going to the website, entering your guide sequences, and then the guide shows up in a couple of days. And these are really intended only for research use for lead discovery, to identify your gene editing strategy. And they're not meant in any way to be used in an animal, for example, or in any kind of preclinical IND enabling study. The IND enabling grade is more of a medium to large scale guide. So this is after you've identified your optimal gene editing strategy. You've kind of nailed down the guide RNA that you want to use for your gene therapy. And so you're going to need larger amounts of this so you can do some preclinical and pharmacology studies, and maybe your GLP tox. So these come with a greater level of purification, traceability, for example. And then once you've done all of those IND enabling studies, you've sent off the IND and you've received clearance to start a phase 1. Clinical GMP is where you would come in and actually get a clinical grade material for what we call first in human or early and late phase, also clinical trials. And so these are actually manufactured in kind of different ways. So the IND enabling material is actually synthesized on the same machines that we'll use for the RUO material, but it has a greater level of purification than an RUO molecule. The RUO molecules don't need a ton of purification. You just need them to work and show you effective gene editing in the lab. Whereas for the INDE, you also need to start to think about toxicology studies, for example. The IND enabling guides are made in a sort of semi-controlled area with traceable raw materials and some documentation. The GMP material is actually made on a qualified, validated oligosynthesizer, also with a high degree of purification, but in a controlled manufacturing environment with fully traceable raw materials and identity testing, and also comes with a full amount of quality control documentation, quality assurance review, and it meets all of the current regulatory requirements. And so here's just a quote from a scientist, Mammoth Biosciences, who was formerly at UCLA, who's really kind of talking to the idea that Synthego has really decided to kind of stand apart from the crowd by just supporting people all the way from the research use directly into the clinic. And this is really helpful and important for scientists developing, so they don't have to switch manufacturers halfway through their process and then re-optimize and revalidate all of their experiments. And actually, speaking of that scientist, Mark, he was actually part of the development of a CRISPR cell and gene therapy that Synthego has supported all the way from the bench to the clinic. And this is another ex vivo CRISPR therapy to cure sickle cell anemia that actually relies unlike the CRISPR Vertex clinical trial that relies on removing the repression of fetal hemoglobin, this is actually a direct correction of the sickle allele in the beta globin gene, an ex vivo therapy where they're using a single-stranded DNA donor to do CRISPR-based homology-directed repair, and then taking those repaired cells and then reinfusing them back into the patient. And so if you're interested to hear a little bit more about how we supported this project all the way from the very beginning, the discovery phase, all the way now as it goes into the clinic with Mark DeWitt and also Don Cohn at UCLA, please feel free to log on to our podcast. And there's a nice interview we did with them recently about this project. It is going into a clinical trial. Don Cohn and also Mark Walters at UCSF are leading this trial. And so it is expected to be able to dose their first patient early next year. Okay, so as I just mentioned, supporting people all the way from the discovery phase into the clinic, how we're thinking about this is how we can scale with developers of cell and gene therapies. And so for our own capabilities, this means that starting off when we first launched our GMP facility, being able to support sort of milligram scale of material, which is appropriate for phase 1, first in human, being able to scale that up to into grams this year, and then hopefully in the future, we can push on and make sort of tens of grams of material that can be used for later phases of cell and gene therapies. So now we talked about the actual materials themselves and how they're appropriate for each section, if you will, of the cell and gene therapy pipeline. I want to talk a little bit about how we support developers, including CIRM awardees, in how they interact with the regulatory bodies, and also show you some of the feedback we've got and help you understand how we're trying to kind of push the narrative on how molecules like this are characterized properly. And so, as I mentioned earlier, we do provide this pre-IND filing and submission support, and also post-IND regulatory support. And so because we're proactively surveilling all the regulatory landscape, we're attending all the regulatory meetings, we're very good at being able to communicate that information back to our clients. And therefore, when we speak to the regulatory bodies, we have a good idea of what they're looking for. It can really help to guide developers through this whole manufacturing process. And so what this means is that we can support people directly through their interact meetings, and then we can provide briefing packages for these meetings as well, and give guidance on regulatory authority feedback. When it comes to the IND submission, we can actually also help to author the drug substance part of the IND submission for the guide RNA for the CMC section. And we have some submission ready documentation. And now we have filed a drug master file with the FDA. So our clients can refer to this drug master file for their IND submission. And then after the IND submission, we can provide guidance and response support to these regulatory authorities, both in the US and abroad, and talk about some of these quality strategies that we've developed based on our CRISPR expertise. And this is just a nice quote here from another scientist who we've worked with at University of California, San Diego, Stephanie Cherky, who is just kind of echoing the point that, like, as you go on this journey, it really is helpful to have people with expertise in this area to kind of help shepherd you along the way. So just some kind of feedback we've had in CMC FDA feedback for the guide RNA drug substance over a lot of phase 1 clinical programs that we've supported. As I mentioned, we've manufactured more than 100 of these batches already, and supported 41 different institutions along the way. We've seen 35 different recommendations from the FDA over these 16 INDs. And typically what they're mostly concerned about are, how do we characterize the purity of a guide RNA molecule, and also understand the impurities that might be present in those molecules. And then secondly, how do we actually verify the identity on a sequence level of that guide RNA? And this means actually developing new methods to understand what the exact sequence is, and how you can use orthogonal methods besides chemical synthesis and sort of molecular weight of a molecule to understand if the molecule that you're going to use in your therapy is exactly what you think it is. So when you think about sort of what are the critical elements here, it's the purity of the molecule, it's the impurities that might be present, it's confirming what that sequence of that molecule is, and then ensuring that you have proper quality oversight and the appropriate documentation that goes along with that aligns to the expectations of the regulatory bodies. And so, as I mentioned, these three different areas when it comes to product quality, the recommendation from the FDA is that a guide RNA has a full-length product purity of at least 80% as measured by HPLC. And so, we can optimize around HPLC analytics to really try to drive improvements of the purification to meet the recommendation. When it comes to impurities, they recommend that all impurities that are present at greater than 1% should be identified as part of the guide RNA characterization. And so we can use things like mass spec to identify what these impurities are. And then also they recommend that sequencing be used as an orthogonal assay for either identity or purity and to quantify the presence of any sequence variants. And so we've been working on optimizing methods to sequence guide RNAs in this way. And so to this point, and as I mentioned, we like to interact with all the regulatory bodies, and we've been part of the National Institute of Standards and Technologies Genome Editing Consortium for the last 3 to 4 years. And so very recently, we took part in a workshop with both NIST and the FDA that was focused on understanding measurements and standards for advanced therapies, particularly CRISPR-based cell and gene therapies. And so we were asked to present some of our thinking and data to this large panel of the FDA, and it really kind of helps us to drive the narrative and make sure that the regulatory bodies understand how we can characterize these molecules and what are kind of the essential components of that. So when we think about looking at guide RNA quality, like I mentioned, there's kind of 3 different ways that we can do that. And we perform some, really kind of state-of-the-art tests to analyze guide RNAs on multiple levels. And so the first would be through HPLC, where we can get a sort of what we call a validated purity measurement. And we can use algorithms to sort of kind of guesstimate what the purity will be based on looking at thousands of these molecules. We can use mass spectrometry as well to actually dive in a little deeper and look sort of more closely under those peaks using HPLC and understand what the different fractions are and identify those fractions and what the potential impurities could be. And then we've developed methods for sequencing guide RNAs using NGS, and we can take some learnings from that as well, because there are many ways of doing this. And on top of that, we can start to observe how different guide RNA sequences can impact this particular type of assay. Excuse me. So when we think about looking at guide RNA quality, like I just mentioned, we're thinking kind of in 3 different dimensions here. So the HPLC, the MS, and the sequencing. And so each one of these comes with different challenges. They're important to understand so you can properly characterize a molecule. So it turns out as you start to chemically synthesize a molecule of RNA up to 100 nucleotides, which is the length of a SP-Cas9 guide RNA. And previous to 2016, when we launched this as a product, was actually very difficult to do. It turns out as you get closer to 100 nucleotides, your ability to make those molecules becomes sort of less and less efficient. And so your yield, as a result, your overall yield of the molecule will actually start to decrease as you get sort of longer along in making this molecule. And on the bottom right here, we can see a bunch of different chemicals that are actually sort of like taking part as part of this, like all of these different side reactions that can occur during chemical synthesis. So this is a natural part of chemistry. When you start synthesizing a molecule, like you do with RNA chemical synthesis, guide RNA synthesis, you invariably will see the development of other species of molecules and chemicals that appear as part of that sort of like side reactions of those processes. And there can be, a large number of these molecules, and predominantly they will be inert molecules that will not show any activity for the guide RNA in question. or sort of can be identified as impurities in the sample. And there's really just no way of getting around this. This is sort of, like I mentioned, kind of a natural byproduct of RNA synthesis, and DNA synthesis is also the same. And so, the regulatory bodies are aware of this. I think everyone who works in this space understands that it's essentially impossible to make a molecule of RNA or DNA that is 100% pure. And so when we think about using a technology like HPLC to measure the quality of the molecule you're looking at, and in this particular example here, we're looking at a molecule of about 72% purity. You can actually see this large peak here, and what you're actually kind of failing to see, and what you're missing, is that under this peak, there can actually be other species of RNA that you may have missed by just looking at purely HPLC. And so in this case, under this curve, under this peak here, we can see an N-1, so that's like a 99 mer nucleotide guide RNA, and N+1, which is 101. And these are important to characterize because theoretically they could perhaps affect your gene editing outcome, right? If they're perhaps changing your 20 nucleotide CRISPR gene editing window. You'll also see some of these like short truncations, and also these kind of longer pieces that could have these chemical adducts to them. And so when you need to take a closer look at these molecules, what you can do is do a fraction collection and then do mass spec to understand what some of these other species are. Unfortunately, the mass spec is not quantitative or a wide mass range, but it can let you look into a small window and actually see more closely what those molecules are for small fractions of those peaks. So we do fraction analysis that allows us to kind of predict the quality of the guide RNA. So in combination with HPLC, fraction collection, mass spec, we can start to build these kind of like three-dimensional like maps of what we feel a guide RNA actually looks like. So just to kind of sum up some of the challenges in characterizing guide RNAs using these methods. As I mentioned, no guide RNA is 100% pure. It's impossible to synthesize a molecule of DNA or RNA that is 100% pure. There are biases in each of these methods that we use, HPLC and mass spec, and that's why we use them together. And they both obviously have limits of resolution as well. So what we do is try to continue to understand what these biases are, what the limitations are in our methods and the methods in general, and seek to improve upon them and also our detection capabilities. So the last part of that sort of three-dimensional way of looking at a guide is to do sequence verification. And so we can use this as a type of analytical assay. So what this needs us to tell us is how the molecule was actually put together in the order that we intended. So perhaps the molecular weight is correct, but the order of those guides, of those single nucleotides in the guide RNA target sequence, maybe they're out of order. Theoretically, this should not happen because when we're doing a chemical synthesis, we're enacting process control. We know which base is being added on next. However, this is something that the regulatory agencies are interested in understanding. And they can also help us perhaps understand if there are any impurities that are present. And so what's really quite challenging is to try to understand if you do one of these types of assays, what the difference is between an actual impurity that is present in the guide itself, or what is an analytical artifact that can result from the generation of sequencing data of a guide RNA. And so, these methods require you to take a purified guide, essentially do some sort of ligation. So you can make a cDNA, amplify this, then do NGS library prep, sequence, and then use a bioinformatics pipeline to kind of deconvolute what's real and what's not real. And so there are many different methods like to start this process. And so they all involve using reverse transcription to make a piece of DNA first. And so you can put ligation sequences on, adapters on either end of the guide RNA. You can just directly reverse transcribe the scaffold itself. And so each one of these methods comes with sort of pros and cons that can lead you to have an inaccurate sequencing result. And so on the left here, what we're looking at is 4 different conditions, four different types of protocols that we've used to sequence a guide RNA. And you can see that when we look at the reads that are aligned to the reference sequence, which is what we think the guide is, we can see that like each one of these actually produces a different amount of alignment. And so what we need to take into consideration is that sometimes secondary structure is present in that guide RNA sequence, and it can lead to analytical artifacts. So, it's really important to have controls and do this a number of times to understand when you might encounter artifacts based on the sequence itself, and what you expect. And so on the right here, you can see in condition X, you're seeing these truncated artifacts that are actually not present in the HPLC data where we have a single peak. So we know they're not real, they're actually analytical artifacts that are arisen from this method. So thinking about using this as an orthogonal method, very important to understand that like you don't want to introduce further complications, bias, and analytical errors based on the method you're using. So we feel confident that we've now developed a method that can be used to give very accurate sequence description. So no assay can account for all the biases that you have. In general, using these processes, reverse transcription, amplification, NGS, will generate analytical artifacts. And so, again, like I mentioned, it really is important to understand what secondary structure looks like. You can do this through the use of sequencing controls. And it's important also to note that something like this can only be used as part of an aggregate with these other measures, like I discussed earlier, mass spec and HPLC. Okay, so that's taken us to the end of the webinar today. I just want to acknowledge some of the amazing team of scientists and engineers at Synthego who worked on generating some of the data and building some of the buildings that you've just seen. And so just to kind of to wrap up, we're really trying to help democratize CRISPR entry into the clinic in the same way that we tried to democratize its use in the research and discovery lab. And as we discussed at the beginning, we're now a CERM industry resource partner. We have significant experience in synthesizing, analyzing, and quality controlling guide RNA molecules, and working with the regulatory bodies to kind of lead this conversation around what purity of a guide RNA looks like. And in addition, I'll say we'll also have significant CRISPR expertise when it comes to engineering cells. So we have a number of subject matter experts who are willing to also provide support. Based on feedback we've had from developers of cell and gene therapy, we've developed these phase-specific guide RNAs that can really help to streamline discovery to the clinic journey and make sure that you're using the right type of material for the right type of study. So how we've developed, how we supported cell and gene therapy developers since 2020. As I mentioned, we've now manufactured more than 100 of these client batches. We've supported 41 different institutions, industry, academic medical centers, from IND enabling into phase 1. We've gone through 20 successful audits and now have supported 16 IND and pre-IND studies, 6 of which have been cleared. So just want you all to think about at the end of the webinar now, how we can best support you at CERM awardees in this cell and gene therapy development roadmap, all the way from discovery up through preclinical into the clinic. And so I'd just like to thank you for your time, for listening. If you like more information on what we can provide and how we can support you, please feel free to visit our website here, put down on the left. And I'll be happy to take a little pause now and take any questions that there might be from the audience.
Okay, thanks, Kevin. That was a great talk. We do have a few questions from the audience. Let's start with this one. So you just spoke about phase-appropriate synthetic guides. Why is it important to use a single vendor for RUO to GMP?
I think it just really, like I mentioned briefly in the talk, I think there's a couple of reasons for that, to stick with a single vendor all the way through if you can. And that is because at least when it comes to using the guide RNA part itself, switching vendors may end up in you receiving molecules of different purity and consistency. And this could affect your ability to sort of like duplicate your results maybe in a cell line in the lab into like a primary cell, or even later into an animal study. So at each step, you're going to have to like kind of go back and re-optimize and sort of like re-qualify the reagents that you're using. So I think, you know, when we talk to clients, you know, they're really interested in being able to go on this journey from A to B, from discovery to clinic with a single vendor. It really helps to make things less complicated, and you know, we can help to kind of guide developers along, kind of remind them when they need to get the next stage of purity, and then also again with our abilities to provide this regulatory support, you know, we kind of always have an idea, you know, what they've been working with.
Perfect. Thank you. Okay, the next question from the audience. Does Synthego have partners that can make Cas9 mRNA and or LNPs?
Yeah, it's a great question. I think everyone's kind of thinking about how do they make a formulation in the most appropriate way. Currently, right now, there are no vendors that are kind of doing all of it together. I would hope that, you know, one day in the future, there'll be a single shop that can make you a GMP guide, a GMP nuclease, whether that's protein or mRNA, and then formulate it into an LNP. So yeah, that's actually just not happening right now. We don't have specific partners, but we certainly have vendors that we could recommend for those materials and for things like formulation as well. So I think, you know, again, eventually the industry will sort of like head to this direction, right? Where we'll try to get everything formulated, put together, manufactured all in one GMP type space.
Okay. The next question, do you know if the FDA has ever allowed a team to use INDE grade sgRNA in their FIH trial?
Yeah, first in human trial. So that's a really great question. So I would probably rather defer that question to my regulatory person, excuse me, but what I can tell you from what I understand is, you know, obviously the use of GMP material is a recommendation from the FDA. And so, you know, if people can make the case that maybe they don't need to use a GMP molecule, they'll have to run that by the FDA. You know, I think there's obviously some risks involved and in doing that. And, you know, it's particularly around the ability to have certain levels of traceability and documentation with the molecule that FDA may ask for. So I think it sort of depends on what the cell and gene therapy is, right? So perhaps if you're engineering an IPS line and then later you're going to make a master IPS line cell bank under GMP, maybe your conversation with the FDA might be, well, I can just use an IND enabling guide to do the engineering in the IPS line because I'm actually going to take that line later and then expand it under GMP conditions. So I think, you know, all of this has to be part of the Interact meeting, or, you know, part of the pre-IND submission. But yeah, I think, you know, I can't speak to that directly if they've allowed this. I'm sure if it's possible to make the right, you know, sort of case that you need to use a lower grade of material, maybe the FDA will go for it. But you know, in general, the recommendations have been from them that even for ex vivo cell therapies, that GMP reagents should be used.
Thank you. A couple of questions to the purity analysis that you spoke of. Can small amounts of N+1 or N-1 species really affect your full-length product performance?
It's another great question, and kind of a difficult one to answer. I think, you know, it sort of depends if those N+1s or N-1s are actually fully functional guide RNAs, or if they contain some kind of like chemical adducts that will sort of make the product inert. So, you know, in general, you know, this is something that can be kind of worked around by understanding what the off-target analysis is of the guide. And so typically, you know, people are choosing guide RNAs for gene editing strategies that will have, you know, a certain number of mismatches in the guide to prevent off-targeting at other loci. So, you know, having an additional base or one less base, you know, if you've done your kind of homework on understanding where your targeting is, likely is not going to be problematic. You know, that said, it's not likely that most of these molecules are going to be active anyway. So, I think, while it could potentially pose an issue, that's why, it's important to understand what amounts those impurities are present in the molecule. And, obviously those things have to be reported.
Okay. Along the same lines, you talked about some of those chemical byproducts of the synthesis process. How do they affect, or how can they affect the performance or safety of the guide RNA? And does Synthego provide a report on these byproducts?
Yeah, so if the byproducts are there at a greater concentration of than 1%, then we will provide an impurity report. I would say in general, they're going to be inert, and so they're most likely safe. And so they should not affect the level of gene editing.
And the last question I have, unless anybody else wants to populate a few more real quick. The analytical work you've presented was performed on 100 MERS. How do these characterizations apply to alternative guide RNA products for non-SP-Cas9?
Yeah, certainly we can apply all of these methods to other types of guide RNAs as well, whether that's for like a Cas12a guide RNA or maybe a Cas13b or something like this. So yeah, all of these analytical methods are kind of sort of like guide RNA agnostic, if you will.
Excellent, thank you. So with no more questions, that concludes our webinar. Of course, you can reach us at synthego.com/GMP. We're happy to answer any more questions that you have and connect you with technical or sales experts moving forward. Thank you, Kevin. Thank you, Lisa. And everybody have a great day.
Yeah, thank you so much for coming.